So first, are there any questions about fish? No questions? No questions about fish? Oh, good on fish. Okay, we'll come back to fish. So today, I'll just sort of go over the cochlea with just one close-up picture to see where these hair cells are. And it's actually, I'll zoom out a little bit. I just showed you one hair cell, and it was kind of hard to see where stuff actually is. So a little bit on the cochlea. Then another major difference. So in the somatosensory system, we talked about there were these discontinuities in the cortical maps in the somatosensory system that you don't find in the visual system because of having to kind of cut up all the, the body surface into a bunch of little pieces and paste it on, onto the cortex. There's another big difference between the visual system and the somatosensory system together versus the auditory system. And so we'll talk about, and it's important to sort of get an idea. It's a real thoroughgoing thing, goes all the way up into the cortex. So then we'll talk about some of the structures in the brainstem. And this is another difference with the somatosensory system. More stuff is going on in the brainstem in the auditory system. So more stuff happens before you get up to the cortex. And so in some sense, the auditory cortex is at a higher level than visual cortex. And so we'll start with the cochlea. So just quickly draw that diagram. We got the ear, and there's a long canal, and then it goes to the eardrum, and then you got like the three bones and the stapes, and then that sort of hits the cochlea, and the cochlea basically, it's got this kind of snail-like thing where it's kind of wrapped up into a snail-like coil, and then it's got the semicircular canals in it, and there's three of those guys and another one sort of going this way, and then it's got the utricle and the sacculus in there also. And so we're going to talk mainly about the cochlea today. That's the cochlea, and then we got the sacculus. That's one of the ones that detects linear acceleration. And then we got the utricle. That's another one that detects, one of them detects two directions and the other detects the third. And then here's the semicircular canals, which should be called the circular canals. And those guys detect angular acceleration, so rotation, so they don't give any response if you just move in one direction. And so there's basically 6 fields of hair cells. There's one in the cochlea, one in the sacculus, utricle, and 3 in the semicircular canals. So let's focus on the cochlea. So we'll focus on the cochlea. So, and we'll talk about the other things later when we talk about eye movements. But the first thing is we talked about sound waves. So here's sound waves coming into the into the eardrum. And so if we back out and say, what is the cochlea mainly doing? And so what the cochlea is mainly doing, it's doing a short time window Fourier transform. It's not an exact Fourier transform, but it's kind of close. And so that was, you probably heart sunk on that. So what does that mean? So a short time window Fourier transform. So if we go back to what sound waves are, so it's kind of an amazing thing when you think about it. The sound wave that comes into your ear is just a one-dimensional signal, if you think about the dimension being time. So it's basically just a single instantaneous air pressure. So I'll draw a signal that has some high frequencies in it and some low frequencies in it. But it's just a single thing. And the amazing thing is that just that single variable, which is just pressure. I mean, it's not a great deal more than atmospheric pressure. It's like, 0.001 more than atmospheric pressure and negative 0.001 less than atmospheric pressure. It's just a small amount of pressure change in the air. But that signal is just a single thing. At any one point in time, it's just one pressure. And now think about a whole orchestra playing or a noisy bar. All that is just squashed down into one variable. And so like a whole song with singing and bass and drums and everything like that, it's just a single thing at an instant in time. And so what does the cochlea do? Well, if we sort of take a little chunk of that and blow it up, so let's just take the chunk, say like from here to here. So just a little chunk of that and blow it up. And so what does that look like? So that looks like it goes up, down, something like that. Okay, so that's our signal for a little chunk of time. And so what is a Fourier transform? So a Fourier transform basically is you need some time. So you take a little chunk of time and you turn it into frequencies. So you get rid of time entirely and you just estimate what are the frequencies over that whole little thing. So that's what a Fourier transform is. And if you take my neuroimaging class, we go over in a lot of detail so you can see how it works. It's a little subtle. It's kind of involves complex numbers, but we'll just leave that out for now. So it's important to get the dimensions. So this is time going that way, and now we turn it into frequency going that way. So now there's no time at all. And so if this was the signal, simple signal, it just has two different frequencies in it. What does that look like when we do the Fourier transform? And if we look at the amplitude of the Fourier transform, because it's complex numbers, so amplitude of ft. We would have some low frequencies in there, and then we have some high frequencies in there. And so what has the Fourier transform done? Well, it's kind of decomposed this thing into, so you can see there's actually a low frequency in here. Here's the low frequency. So it's decomposed this thing into a low frequency, and across all time, and a high frequency across all time. So that's essentially what the cochlea does. Now, it can't do it instantaneously. It needs some signal spread out over time. And then what it does is it takes these little chunks of signal, and then it turns them into a frequency spectrum. Now, over time, now, here's one little chunk, and then, the orchestra's complicated sound. Everything's changing over time. And we're talking little chunks of time here, like maybe a 50th of a second, something like that, small amount of time, or a 30th of a second. So a tiny little chunk of time, but still some time. And now we go to the next chunk of time and do the same thing, and the next chunk of time and do the same thing. And so what will happen at a later point in time, well, this spectrum, so this is a frequency spectrum, will change, and that's how you actually hear things, and hear that the bass came in, or hear that somebody hit the cymbal, or somebody said something, they said a T. So that's essentially what the cochlea is doing. It's doing a short time window Fourier transform, which means take a little chunk of time, make sure this is time, take a little chunk of time, here I blew it up over here, a little chunk of time, and turn it into a spectrum of frequencies, and then keep doing that. So how does it do that? So it mainly does that with the in a mammalian cochlea with the mechanical tuning. And so what is the what is the mechanical tuning? Let's draw sort of nice big, it's big cochlea. Okay, so you got a big cochlea. So if you take apart a cochlea and just section right through like one of those turns. So we're just looking at one. So here we're sort of expanding out just one turn in that cochlea. And I need my cochlea to be a little bit wider here. So, okay, that's nice, big cochlea. So if you look at that, you'll see there's one cavity at the top, and then there's the basilar membrane. So let's put the basilar membrane in here. So that was we said that was kind of a rigid membrane there, and then the hair cells are on top of that. And how do they sort of sit on top of that? Well, it sort of comes up like this. This is solid stuff, so solid tissue. This is fluid right here. So I'll have to continue drawing for a little bit. And so this kind of comes like that. There's a little thing that comes there, and then it comes around, and then we've got the tectorial membrane. So here's the tectorial membrane, and then it goes down and then up, and then here's where the hair cells are. So last time we were just looking, so let's put some hair cells in here. So here's the one hair cell that's the main frequency business. And then there were those pictures that I showed you where there were 3 cells that we'll see, 3 hair cells here. And they're basically touching this tectorial membrane. So here's tectorial. And this guy is basilar membrane. And so we said the way this works is the tectorial membrane sort of goes up and down. And why does it? So the waves in the tectorial membrane are sort of coming out of the board or into the board, because this whole tectorial membrane is kind of coming out and spiraling around. So there's another cavity underneath here, like that. And then we mentioned that there was the ganglion cells. So the ganglion cells are kind of sitting right next to this, and those are the guys sort of going into the CNS. And that's called the spiral ganglion because it kind of spirals around, just like it's on kind of the inner turn there, spiral ganglion. That's what the ganglion cells in the auditory system are called. And this comes out, and the axons run along, and then when they get to these guys, they make a little tiny mini dendrite that will receive a synapse from that hair cell and send spikes in. So these guys are called the inner hair cells. Inner hair cells. And these are the outer, naturally the outer hair cells. And so how does this, how does the sound waves get in here? So they come in through the oval window. And what is the oval window? So the cochlea, this stuff is pretty hard. So this this tissue here is kind of like it's like a rigid tissue. And it's all rigid except for the, for the oval window. So this is all a pretty rigid thing. So the oval window is right where the stapes touch. It's like a little thin membrane that is flexible. And there's another window. There's the round window. So this guy's the round window, which is also flexible. And so that way you can sort of touch the oval window and it will cause the round window to move. So this is the oval window. That's where the stapes attaches to. And so what happens is basically this upper cavity is attached to the oval window. So this guy is it's sort of going in there, or out as the case may be. No, it looks in. Yes. Okay. So this is basically, from oval win stapes. And then that, if you look at so how did this, get started off? It's easier in something like a bird. So if you look in a bird, the cochlea is called the legina, but it's the same thing. Here's that basilar membrane right there. And so what happens is, here's the oval window. These fluid cavities are attached like this. And so here's the round window. So you have sort of a continuous fluid cavity that goes to the end of the cochlea and then connects back up to the oval window. And that's the same thing that we're looking here. So what we're looking here is just in cross section. So here's all the, all the hair cells are, mounted right, inside here. So here's, there's the hair cells there. It's the same way in a bird. And so basically here we're sort of, here the fluid is sort of, coming out. Coming out. So this is, two round window. And when you look at something like this, it's just, it's just mind-boggling to think, like, how many genes do you have? You've got 20,000 genes. How many genes does a bacterium have? It has about 6,000 genes. How many does a fly have? Maybe about 14,000. So it's almost the same number of genes. We only have 3 times as many genes as a bacterium. And somehow those 20,000 genes can make this. It's just unbelievable. It can make all these, somehow signals how to make all these cavities, put all the hair cells in the right place, make different kinds of hair cells, make a tectorial membrane. How the heck do you code that into just 20,000 genes that are coding for 20,000 proteins, basically? It's just mind-boggling. It's biology. It's just mind-boggling. The amount of specificity that you can get from a relatively small amount of, relatively trivial amount of, genetic information, just 20,000 genes. So, okay. So that's the cochlea, and it sort of, goes up and down, and then the hair cells go back and forth. It wiggles the hair cells. Because basically when this thing moves up, there's a little shearing from the tectorial membrane. So what about those hair cells? So the inner hair cells are basically the main frequency signal. So they're the ones that are actually sort of mainly giving you information about different frequencies that comes into your sensory system. And these guys are myelinated. So they have very good conduction velocity. And they can basically sort of indicate essentially every time up to a pretty high frequency that the basilar membrane goes up. So you'll give a spike, a timed spike, just as it goes up, as the hair cell gets tweaked. A single spike is exactly timed to the peak in sound pressure, up to, in the case of owls, up to about 5,000 cycles per second. So they're myelinated so that they have a good response time. These other guys, so that's the inner hair cells. The outer hair cells are the ones that are electromotile. So these guys actually, I said, like, some of them you don't even have to electrically stimulate them with a synapse. You can just play sound to them and they'll like bend, like little people will bend. But these guys also receive input from the central nervous system. And so there are some, so these cells are synapsing on spiral ganglion cells and going into the central nervous system. But in addition, they actually have some cells in the central nervous system that project out to the hair cells. And so they come from the peri-olivary nuclei. And so these guys actually come out of the CNS. These cells are going to the cochlear nuclei. Those are the input, input cochlear nuclei. We'll talk about those down here. But these guys are actually sending, these are neurons here in the central nervous system. So here's CNS. And they're actually synapsing back out onto the actual, especially to the outer hair cells. So that's a synapse that's going in as opposed to this synapse which is coming out. So these guys are electromotile, so the central nervous system can cause them to actually contract and condition the signal. And so these have been called the cochlear amplifier. And so the idea is that they kind of, especially through some signals coming out of the central nervous system, they actually condition the signal, and so they get, olivo-cochlear bundle. That's those axons that are coming from the central nervous system and synapsing on the hair cells, olivo-cochlear bundle. And these guys are unmyelinated. The input fibers are unmyelinated. So the outer hair cells and the inner hair cells are quite different from each other. So this guy, the inner hair cells are the main frequency signal coming in. These have the effect of amplifying the signal, and this setup where you've got the central nervous system receiving things but sending something out to the detectors, it's kind of like the muscle spindle muscles. So if you remember the muscle spindle muscles, there's a detector. There's a detector which is detecting stretch, but then there are the gamma motor neurons, and they project out to the little muscle spindle muscles. And so it's conditioning the signal. And when we actually looked at how it worked, it turns out what actually makes the signal is actually it's the gamma motor neurons that are causing the little tiny muscles to contract in the muscle spindles, and that's what's actually stretching the muscle spindle when you get an unexpected load. So the same thing is happening here. So the central nervous system is kind of directly controlling the detector and adjusting the detector to for conditions, like how loud is the sound, what frequencies does the sound have in it. Now, that doesn't happen, it doesn't happen in the visual system in mammals, but it does in birds. So if you look in birds, there's a big nucleus that actually projects out to the retina, and it actually touches, it synapses on cells in the retina and sort of conditions how those cells receive visual inputs that are sent back into the nervous system. Okay, so it's complicated, but very high performance, all in the service of doing this kind of thing, which is essentially taking small chunks of sound over time and turning it into frequency, physically turning it into frequency. So different parts of the basilar membrane, the hair cells at different parts in the basilar membrane respond to different frequencies. Okay, so any questions about the sort of cochlear review? Hair cells, various things can sort of take out your hair cells. Some chemicals can take them out. That's kind of a nasty.
Yeah. Why are inner and outer hair cells called what they are?
Yeah. So inner means inner relative to the axis of the snail. So basically, the way the snail works is it's going around sort of this way like that, and so this is the inside of the snail. That's why it's inner. Yeah, I know I had a trouble sort of remembering that. And so this is the outer direction. This is the outer direction in the snail. Yeah, so that's why they're called inner and outer. It's from taking apart some of God's creatures, seeing where stuff is. Okay, so any other questions about the cochlea, the amazing cochlea? Yeah, sometimes viruses can get into your cochlea. It's can be a problem, cause tinnitus. Yeah. Yeah. Oh, ear infections, yeah. Well, yeah, so that is, ear infections, sort of chronic. I had those as a kid. There's a lot of, if you get inflammatory, especially various kinds of inflammatory problems that block some of the normal fluid flow in the ear canal, you can get these chronic ear infections. And I used to get many of them. It would pop my eardrum, actually. I would get an infection in the middle ear where the bones are, and I would get all gunk in there, and eventually you'd build up the pressure until it actually popped the eardrum. It actually surprisingly doesn't damage hearing that much. You pop your eardrum, all the gook comes out. Eventually your body sort of cleans up all the junk, and then it puts the eardrum back together, attaches the stapes back up to it, and you're good to go. That probably happened like 6 or 8 times when I was growing up. But obviously, that the tendency toward some kind of inflammatory, problems in your hearing could spread to other parts of the body, and could easily have a correlation with psychopathology or other things. Well, I'm completely sane, as you can see, totally sane. But yes, no, it's definitely could definitely be associated with some kind of inflammatory attack on the brain, for instance. Okay, so let's go over to 1D versus 2D. So this is a very simple idea, and it has to do with the fact that there's this fundamental difference between sort of the vis and somato systems. So in the visual and somatosensory systems, you've basically got a two-dimensional receptor sheet. So here's, it's got a little thickness to the retina, and it's got some layers in the retina, but we can sort of approximate that as a sheet that has, 2 dimensions. So 2D receptor sheet. Now when you go into the brain, the brain is, the retina is 3D too, but the brain is really 3D. So if we go into the geniculate, what do you have in the geniculate? Well, you've got a map. So, there's a map going this direction in the geniculate, 2D map. But you've got another dimension in the geniculate, all the layers in the geniculate. And so what happens is that a point in the retina will basically project to a little tube of cells. So here's our little cube, 3-dimensional cube. So basically what will happen is here's a little tube of cells in the geniculate, all the different layers in the geniculate. And what happens is a point in the retina comes out and, let me make that one a different color. It comes out and essentially projects to a line. So when you look at this sort of geometrically what's going on, you've got like a point here in the retina and it's projecting out to a line, little line through the geniculate. It is like a little tube. If you look at you'll see a little set of labeled cells in a tube. Now, if you go to another point in the retina, what's that going to do? Well, that's going to project to a line somewhere else in the map. So don't overlap that guy exactly. So here's another one, another little tube. And so this other point will, project out to a different little tube. And when you look at higher level projections to the superior colliculus, or if you go to the cortex, you see the same thing. So like one point in that little tube will basically, project to a little line in like a little column in the cortex. So you see the same thing. And so we could call this, point to line kind of projection. And the somatosensory system is the same way. So when you have a map, you've got a map in these 2 dimensions on the surface of the cortex or the surface of the superior colliculus, but then there's a whole bunch of different cell types as you go down in depth, but they're all sort of servicing the same point in the map. So this is like area 1, area 2. So that's true in the visual system and true in the somatosensory system, because the skin is the muscle is a little more complicated, but the skin at least is a straightforward 2-dimensional sheet like the retina. So what happens in the cochlea? Well, it's quite different. You've just got a line basically. So at the high frequency end of the cochlea, you've got the inner hair cells and the outer hair cells, but it's basically just a 1D receptor sheet. It's 4 wide, but it's basically 4 is close to 1. So it's a 1D receptor sheet, receptor line. It's just a line of receptors. And so now what happens in the auditory system, it's a little more complicated. So what happens in the auditory system, you have an extra dimension. So if you look at what happens when these projections come in, they come in and they actually project to a plane. So here's a little plane. So they actually hit a plane. And when you actually look at the, so it comes in and it actually sort of spreads all over this plane. So it's a real thing. So a lot of times if you look, if you read any auditory neuroanatomy, they'll say that in the auditory system you have these projections to a line, but that's kind of a line when you've cut. It looks like a line if you just cut through this three-dimensional structure, but if you reconstruct the whole thing, what you have is like a little sheet, a sheet of projections. And if you look at a different frequency is going to go to a different, a different plane. So here's another plane. Another little plane. And so this different frequency is going to go to this plane instead. And so the bottom line is that you'll have just a 1D map. Now there's another dimension in the cortex, so it's 1D. Now there's another dimension, and we'll see that the brain uses that dimension for other things. But there's only one map dimension, because you've just got a line of receptors. So that's a fundamental difference between the processing stages in the auditory system and the visual and somatosensory system. And you see the same thing happening when you look at the next layer up. So what you'll see is there'll be, let me put that plane just a little bit over so that the lines don't intersect with each other. Yeah, so you see the same thing where basically a line in the, a single point in one area will project to a plane in the other, in the higher area. It isn't a plane exactly, it's like a bent little potato chip or something like that. And so you can imagine it's a little difficult to visualize these things, but this will go and then project up to a plane. And this happens throughout. So this is area 1. Area 1 projecting to area 2. So this continues as you go up. You go up in the system. So it's a fundamental kind of architectural difference between the visual and somatosensory system and the auditory system. So this is, forgot the heading here. Auditory. Visual and somato. Is there a question or? So there was an experiment in which they did bad things to ferrets, where they tried to make the visual system grow into the auditory system, just to sort of examine how map formation is occurring. And so they got a ferret and they essentially caused the visual structures, like the superior colliculus, to actually grow into the auditory system. And what happens is they make a map there. These visual things will make a map in the auditory system, but when you look at the map, what does it look like? It looks like the auditory system. And so it said, like, I don't care if you're from the visual system, you're going to make planes, because that's what we do in the auditory system. And surprisingly, the ferrets could actually use their visual system, even though somehow they had made planes. They could use this information coming in from the retina and then going into the auditory system, doing the auditory thing and making planes, and they could actually see with it and actually do spatial vision with it. Yeah.
Is there any alterations to vision?
Oh yes, no, they weren't. It clearly wasn't. You don't do this at home. It's bad to connect your auditory system up to your visual system up to your auditory system. It's better to hook it up to the visual system. Everything works better. But it is that sort of that's a clear demonstration that the auditory system wants to do this. This is kind of how things are done in the auditory system. Okay, so any questions about that sort of 1D versus 2D? So it's another difference. So you still have maps, but you've only got sort of one receptor dimension, and then you've got some other dimension, and we'll find out there's a whole bunch of things that are different things that are mapped in that dimension that's perpendicular to the receptor dimension. So it's not like it goes to waste. But the fundamental difference is that the signal coming in is a 1D signal versus like a 2D signal. It's just a signal from different parts of this line of hair cells, as opposed to the retina, where it's coming from different points on a two-dimensional sheet. Now, it doesn't mean that, we'll find out that the auditory system can actually make a two-dimensional sheet. When we talk about like the making an auditory map of space, you need 2 dimensions, and the auditory system can do this. But it's starting off with 1 dimension. Okay, so that's the just enough time to go to the auditory brainstem. So now it's just a mess of names of structures, and they have their names are that's the detritus of history, how things got their strange names, but let's give myself a little more space here. So we'll make a nice fat brainstem. Okay, okay, so first we have the pons midbrain junction, of course. So this is the pons midbrain, to sort of put things in general perspective, and then I'll just dash it so it doesn't mess up our. So here's the fourth ventricle. So here's the midline. And the auditory system, you probably know there's connections that go back and forth between the ears in order to compare signals on the left and right ears, which, break the rule of Serino. But most of the connections follow the rule of Serino, so we'll draw those out. So if we start off, the cochlear nuclei are located in the pons, basically just behind the pons midbrain junction. And so they're not square, but we'll just draw them square for illustrative purposes. So we got 3 cochlear nuclei. And so those are the things that receive the information from the cochlea. So here's the cochlea, and there's a hair cell in the cochlea, and then here's a little synapse it makes. Here's the spiral ganglion cell, and that comes in, and then traditionally, if you read a neuroanatomy textbook, it says it synapses in lines across the cochlear nuclei. But we know, in fact, they're planes. They look like, if you cut a section, they look like a line of synapses, but if you do it in 3D, you can see there's a little shell, little potato chip of connections. And what are those three nuclei? Those are the So the first one is the anterior ventral cochlear nucleus, known as AVCN. So that's the anterior ventral, toward the stomach, cochlear nuke. Okay, so that guy is, and then we're going to talk about owls, and unfortunately, owls have a different name. So this is, I'll say equals nucleus magnicellularis owl. So next time we'll talk about, there's a lot of study of owls because they got such a rockin' rockin' auditory system. They have a good visual system too. And so it's equivalent to nucleus magnicellularis. And if you're thinking now, magnicellularis, that's large cells. This is basically like the Y pathway now remember, it's kind of like the retina. You start off with just one hair cell, the inner hair cell, and you immediately turn it into two different pathways, an X-like and a Y-like. And so sometimes this is called the time pathway because it has sort of a better representation of the exact time of occurrence of a sound wave. So that's one. That's the anterior ventral cochlear nucleus. And this guy is right here. And then we have the posterior ventral cochlear nucleus. That's posterior ventral coch nuke. And that's the X-like one. And so that basically equals something called nucleus angularis in an owl. It's too bad they didn't use the same name. And this one is basically like the X-like pathway. But in the auditory system, instead of calling it X, they call it the amplitude pathway. Now, if you think about it, that's kind of like an X cell. Like an X cell in the retina or the LGN is good at representing how bright something is, because it will continue to respond if you leave the light on, whereas the Y cells, they just respond to the change. And so it's the same distinction, like the nervous system has done the same thing. It takes one signal and it turns it into a signal that registers sort of changes, and then another copy of the signal that registers kind of amount, the amplitude. So this is the time and the amplitude pathway. So this guy is right here, and it's receiving one of those little planes. And then there's a third one, which is a bizarre one. This one doesn't occur in birds, so there's no owl equivalent. It's called the dorsal cochlear nucleus. And this one, bizarrely, it's like a piece of the cerebellum. So it's biology, and the cerebellum is pretty close. The cerebellum sort of goes right over the top right here. So it turns out the dorsal cochlear nucleus comes from the same cells that make the cerebellum. And so it looks like a little weird mini cerebellum. When we talk about the cerebellum, there are these very large cells called Purkinje cells. There's cells in the dorsal cochlear nucleus that look like Purkinje cells. So it's just like this twisted biology idea, like, well, we got some cerebellar generating machinery here, and so we just needed whatever that stuff does. We don't know exactly what it does. We'll just sort of take whatever function that cerebellar machinery can do, and we'll paste a piece of it into the auditory system. So that's the dorsal cochlear nucleus. That has a tonotopic map in it. So if we look at a different ganglion cell, you'll find that it will come in and sort of synapse on a systematically, different part. So you'll end up with a tonotopic map, going across these structures like that. So, but just a 1D tonotopic map, and so then you've got another You got another dimension to play around with. Okay, so now what happens? So one of the things that we said about the auditory brainstem is that there's more stuff going on here. And what's the rationale for that? Well, auditory signals are much faster than visual signals. A good example is dating myself. Back when you would whip out the film projector and turn it on, you'd hear this like sound. Now think about that. You're looking at the film projector and you're seeing smooth movement, but your auditory system is hearing every single frame. It's hearing every single tick, 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 tick. It's distinguishing those ticks. You can actually hear the individual ticks of the frames. Your visual system cannot see the individual ticks of the frames. It just sort of sees some movement, but your auditory system can hear it. And so it's faster, so it's sort of a higher temporal performance system. And because of that, you can't lollygag around sort of like transferring up to the thalamus, long axons. You got to do more work down in the, right down in the brainstem. And so there's a series of structures down here that there's one that kind of looks like an S. And then there's another one over the top. I've drawn them a little bigger than they are. And there's another one that kind of looks like just a flat plate, and there's another one down here. And then there's another one sort of sitting right about here. And then there's this strange structure, which looks like a bow tie. It's got a lot of axons that are breaking the rule of Serino and crossing. And strangely, this thing is called the trapezoid body. Now, that's another one where these guys have to straighten up their names. It's actually doesn't look like a trapezoid. It looks like two trapezoids. This one's a trapezoid, but there's another trapezoid. It's like a bow tie. So that's the trapezoid body. And what is this thing? This is the medial nucleus of the trapezoid body. It has a little map associated with it. And this guy over here is the, don't get these confused. So this guy over here is the lateral superior olive. So what's the olive business? The olive business is just that if you just take apart a brain, you'll see a little bump. Sort of looks like an olive. And so there's an inferior olive, which we'll talk about when we talk about the cerebellum. This is the superior olive, the lateral part of the superior olive. It makes a little bump on the brain, and so that's why they call it that. So that's the LSO, London Symphony Orchestra, lateral superior olive. And so that guy is that one. And then there's there's another map. Dorsal, peri, olivary. That's this guy, and this is our ventral, peri, olivary. You don't have to remember this one. And then this one we'll talk about quite a bit. This one is the medial superior olive. Because that one, it's a medial superior olive. And this one is like the owl nucleus laminaris. And so we're going to talk about that, how this one works. So if we go up into the midbrain, we've got another set of nuclei there. And then we have the inferior colliculus, another big nucleus, and there's some nuclei that are kind of sitting around it like that. There's another one over here. Now there's the superior colliculus, which is above it. I'm just showing, there's two bumps in the midbrain. So this this guy is the inferior colliculus. It just means the inferior little hill, and that's the central part of it. And there's one that we're going to talk about for the owls. This one's called the inferior colliculus X for external. That's one that actually has a map of space in it. And I didn't say what these guys were. So these guys are the nucleus of the lateral lemniscus dorsal part. So that's nuke. So remember, the medial lemniscus was the one that is the projection from the dorsal column nuclei, like the cuneate nucleus, up to the ventrobasal. So that was the medial lemniscus. This is the lateral lemniscus, just because you can just see this big bundle of fibers, dorsal part. And so this one is the nucleus of the lateral lemniscus, intermediate nucleus of the lateral lemniscus, ventral. But they're in the midbrain. They're past the pons midbrain junction. And then last but not least, we get up to the medial geniculate nucleus. Got several parts to it. We'll talk about that later. But this guy is the medial. And remember, the lateral geniculate nucleus was the one that was for visual. So this is the medial geniculate nucleus for auditory. But look at how many parts you got down here. Way more than in the visual system. So you got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19. You got 19 parts before you get up to the cortex. So the medial geniculate is what goes up to at the very edge of the board. The medial geniculate is what goes up to the auditory cortex. And so what are the connections, in this? Well, they basically follow the rule of Serino, most of them, like 90% of them. So there's tons of projections that come down here that come from the cochlear nuclei down into these olives, various olives, medial nucleus of the trapezoid body. And then these guys all have crossed projections, for the most part, up to those areas in the midbrain. And then the midbrain it's past the pons midbrain junction, so it basically has the same side projection. So basically stuff comes in, we're behind the pons midbrain junction, synapse is on the same side, stays on the same side because we're still behind it, and then for the most part transitions over to the opposite side for the midbrain and thalamus and cortex. Now, there are connections going through the trapezoid body that have to do comparisons between the left and the right ears, but those are numerically small compared to this overall picture. And the same thing would happen if we looked at the right side. So you would see come into the cochlear nuclei, go down to the olives and all the stuff, and then go way over to the midbrain on the opposite side. And so it's still kind of, and it actually sort of, if you're doing localization, it divides the world into two. So the left midbrain is dealing with left space. The right inferior colliculus and cortex is dealing with left space. So it's like the visual system where if you're past the pons midbrain junction, the right side of the brain deals with stuff on the left. So that same thing occurs in the auditory system. Strangely, even though it's completely different, completely different modality, completely different. It has to construct space. Doesn't have space at all. It just has frequency. Okay, so I went over as usual, but I made the drawing big enough so you could sort of see the different parts. So what's the bottom line here? Sort of more happens in brainstem, in auditory. And so that implies that, auditory cortex is at a higher level. Than, vis somato. So more stuff has been done to the signal before you get up to the cortex. There's, of course, stages in the cortex. We'll see that there's multiple stages in the cortex, just like there are in the visual system and the somatosensory system. But you're starting from kind of like a more processed starting point. Okay, so any final questions on the 19 structures in the brainstem going in the auditory system? There was a lot to chew on there. So, but like I said, there's just a finite number of parts, and they haven't changed. That's the best part. They haven't changed. They'll be the same 100 years from now if there's still scientists around 100 years from now. So that's the good part. That's the good part. They haven't changed. So it's a finite target. There's kind of a lot of stuff in it, but it's not changing. Okay. Any questions about auditory? So 2D versus 3D, 1D versus 2D made sense. And
So just want to understand basically the pathway and the main components, right?
yeah, so I won't bust your chops for the nucleus of the lateral lemniscus intermediate version. And what we want really is like the overall picture, like you come into the cochlear nuclei, they've got this point-to-plane business. We start to do some comparisons between the ears here. We'll talk about that, how that actually works, because you've got streams coming in from the two sides, and they're spiking differently. And these structures down here are sort of doing the left-right ear comparison. But then we transfer it to the opposite side of the brain and do more stuff up here, and we'll talk about like how do we make this map of space, because you've just got frequency here, it doesn't have any space in it at all, but you can use your auditory system to figure out something's coming from over there. And so how do you get space in the auditory system? So we're going to talk about like the actual sort of computations where you actually figure out how to construct space out of this auditory signal. So that will be the main things to remember. Like, how do we sort of do the computations that get us ready to construct space? How do we actually construct space?
Is the auditory one point-to-plane induction?
Yeah? Oh, I didn't, good point. Yeah, so this one is point, I forgot to label it. No, this is point to plane, yuck. I drew it, but I didn't label it, yeah. So, yeah, definitely. So here's visual system. Let's put some final, there we go. Much better. Yeah, so the auditory system is always point to plane. The visual somatosensory are point to line. thank you for that. Feel free to interrupt me. If I don't see you, just like wave your hands and I'll stop. Okie doke. Think we're good to go.